one of the focuses, we call it the Frugal Science Academy, is essentially going to high schools and bringing synthetic biology to high schoolers and to make them understand that from the get-go, sort of like how computing sort of took off. The high schoolers do this big competition called IGEM. It's an international conference. They make organisms do synthetic biology. Last year, Lambert High School built a test for Lyme disease using synthetic biology. And they're high schoolers. They were on 60 minutes. Essentially, the idea is okay, let's get high schoolers excited about synthetic biology. This is the next big frontier. And one of the issues is that high schools often don't have a lot of lab equipment and they cannot afford like the really fancy stuff that universities can. And so we try and build one by one equipment that's useful to them. And one of the key pieces of equipment there was a lyophilizer. And so a quick overview on lyophilizers. Essentially, you freeze your sample and then you put a vacuum on it, and that pulls out the solvent, which could be water, it could be something else. But for synthetic biology, the key is that with this technique, you can actually preserve organisms. You can you can store them and then you can just transport them at room temperature if you want, if you do it correctly. Except, of course, like the standard equipment is like $20,000 plus, and that makes it a little hard for a high school to afford. And so, like, can we make a really affordable version of this? And so, the vision was let's make a frugal lyophilizer. So, the lab has made many different kinds of things, including 10-cent ultracentrifuge that spins at 120,000 RPM, a $1 electroporator, which allows you to put DNA into cells. And so these things normally cost like orders of magnitude more than that. And the idea was, okay, let's make it accessible. And the problem started with how can we make lyophilizers accessible to the general public? And then so we were building that. And the evaporation challenge came from that question.